everyone. My name is Pietro. I am a PhD student here at the University of Kansas. Weirdly enough, I am a Brazilian studying lizards from the Caribbean in Kansas, and I presented at a New Zealand Congress. So the world goes around pretty quickly. But I wanted to talk to you about what I presented when I was awarded this amazing scholarship by the World Congress of Herpetology. And it's one of the chapters of my thesis where I'm trying to establish the genetics and the genomics of just lizard pigmentation and try to establish this as a study system to study many things such as adaptation and speciation. So right here, you can see my favorite lizard and arguably the greatest lizard in the world on a very biased perspective. This is Anolis disticus. I know there's a lot of hating towards Anolis because they got the first genome, but they're actually kind of nice little animals. Hopefully I'll help you guys see what I see in them. So what I'm doing in my thesis is kind of this integrative approach where I'm gathering data from multiple sorts. So I'm gathering genetic data, I'm gathering genomic data, RNA, but I'm also looking at phenotypic data in particular that I'm interested in. And I'm coupling all of this together to try to identify some genes, some candidate genes responsible for what I find to be very attractive about lizards, which is coloration. Lizards have this amazing colors and patterns that we're all very much used to, but most people really don't see that beauty in them. And dewlaps in Anolis and in many other animals is one of these examples. So my aim and what I presented on in the World Congress was to identify candidate genes and potential gene sets. And by gene sets, I mean, think about a pathway, right? We all hear about the carotenoid pathway, which, you know, gives us yellow colors and orange colors, and we know as humans it's good for us to eat because it helps us with vitamin A, for example. And these animals use carotenoid pathways, so gene sets that make the carotenoid pathway and other pathways to get those colors that you're seeing, like in this little friend of mine. So what I did in a nutshell and what I presented on is one of my steps in my thesis was to do an RNA-seq experiment. So what I did here is that the first animal that I showed you is an orange dewlap, Anolis disticus. So what's interesting about disticus is, so Anolis originally were kind of divided according to their dewlap colors and patterns. Humans looked at Anolis, and a lot of them, when a human saw different dewlap colors, they assumed they would be different species. What we know now with genetic data is that that might not necessarily be true, and disticus is one of these examples. So disticus, we have orange dewlap populations on wet environments and yellow dewlap populations in dry environments, and when they come in contact in these contact zones, you actually have guys like this one that you can see at the bottom of my slide. This is a hybrid. So this animal has an orange kind of center of the dewlap and a yellow margin around it. So what I did here is that I excised the dewlap of animals. Obviously, we went through the euthanasia and everything. And I excised the dewlap of these animals. And I got both pure orange dewlaps, pure yellow dewlaps. And I also got the yellow margins and the orange centers of guys like this. And I basically tried to answer the question, are we seeing different genes being expressed in yellow versus orange dewlaps? So if we assume that they're being that these genes that are being expressed are responsible for the coloration, we would expect different genes to be expressed in different phenotypes. What you can see here at the right is this kind of weird looking graph. What you have in the y-axis is how much the expression of a given gene changes. So each little dot is a gene, a measurement for a gene between an orange tissue and a yellow tissue. And on the x-axis, you have this mean of the normalized counts. This is us trying to account for the variation. So as you go from one side to the other, you're going to get genes that are more expressed or less expressed. And then in the y-axis, you're going to see differences between the two different tissue types. I circled one gene in particular here, SCARB1, also known as CLA1. This is a gene in the carotenoid pathway. This would be one of the genes that we would expect to be associated with this dewlap color. And what we do see is that this gene is overexpressed in one color relative to the other. There are many genes that came out in this approach. As you can see, there are many different red triangles here. And what we're trying to do is kind of hone in onto the best candidates. What I can also say is that we did a pathway analysis where we're basically asking if a whole group of genes is being more expressed in one phenotype rather than the other. And we also found that both the pterin and the carotenoid pathways are more expressed in orange dewlaps. So what I'm hoping to do in the future is to complement this RNA data with chromatographic data. So chromatography, for those of you who haven't done it, is when you try to identify the molecules that are responsible or there are within a given tissue type. So my aim is to do chromatography and histology to try to see what are the cell types in orange dewlaps and in yellow dewlaps, and what are the kind of pigments that are being deposited there. It's tricky because we know that both carotenoids and pterins, which are the other color genes, can be responsible for both red and orange colors. So I'm hoping to use this RNA data, some genomic data in the future, and also this phenotypic data to kind of figure out what are the best possible genes that could be explaining this phenotype that we're looking at. Just wanted to give a shout out to everyone in the University of Kansas, my sponsors, everyone in the Dominican Republic, which are great collaborators, and to the World Congress of Herpetology for the opportunity of presenting this work. Thanks so much. Yeah, so the whole Congress experience was honestly amazing. I would arguably say that when I got the World Congress response saying that I had gotten the scholarship, my neighbors probably thought I was insane. I started kind of yelling and just because I didn't really think I was gonna get it, and I was so very much happy because I had never been to New Zealand. And this was a particularly important moment for me to be out there. And you know, now with the corona, it's even more because I am in my last year of my PhD. I'm starting it right now, and this would be the summer for me to work my postdocs, basically trying to do the networking to try to, you know, figure out where I could go or people that could be interested in my work. And one of the reasons that I wanted to go to the World Congress of Herpetology is because some of the people that I'm interested in working with are not necessarily close to my institution. They're in California, they're in the northeast of the United States, but also they are in Europe. And I also wanted to talk to some people from Australia. So the World Congress was this amazing opportunity that I had in January before all of this happened to just talk to people and see if they would be interested in the science that I was developing during my PhD. So the whole experience was amazing. I was able to, I have some friends that live in Oz that we were master students together in Brazil, and now they're in Australia. We're able to see them and get to know people from their lab. And you know, going to the joint meetings here in Brazil, in the United States, it's different because you end up only seeing people from the US. And even though I try to get out a little bit and go to Evolution where we get people from abroad a little bit more, the World Congress is a whole different ball game. It is, you know, it really is people from all over the world. It really is people that, you know, you read those people in those papers and you didn't really conceive that would be possible for you to see them or talk to them because they live, you know, in a different hemisphere or a different continent. And it is so cool. It was so nice. And the experience of the Congress was just, the bar for the next Congresses are crazy high, because I would say that arguably I ate better there than I eat when I'm here at my house. But that is, you know, that's minor. The space was great. The presentations was great. I love the way that things were divided. So I would kind of like be going around and going to different talks, because you'd have talks on physiology, you'd have talks on conservation, you'd have talks on genomics, you'd have talks on adaptation, landscape genetics, all those things that really, if you end up going to, you know, a smaller Congress, even though you do have amazing things in smaller Congress, is nothing like this. It's nothing, you know, in this dimension. So the World Congress was really, you know, it's the first time I've been. I had friends that went to the one in Canada which they said it was absolutely unbelievable. And since I heard about that one, I wanted to have the opportunity to go to a different one. And I am so happy to having had the opportunity to go to this one that I'll go out of my way to try to go to the next one. And then this picture here is what we did after. So New Zealand is such an amazing country. It has, I would argue that there is something unusual about it, which is the silence in the forests. And I understand why. I understand that it's because you do have, invasive species, and one of the hypotheses is that these little, ferrets and stoats and these little mammals went around eating every bird that would chirp, everything that would be loud. And it is noticeable. You go around these forests and you don't really hear, a lot of birds. So you just have this magnificent landscape, this crazy topography. The country's so, topographically variable. I mean, I didn't even go to the North Island, so I just stayed in the South Island, but it's so crazy cool that, I just kept imagining seeing gigantic moas walking around, a few thousand years, and how that would have felt. And it is a shame, and it is a reminder that, we do need to preserve what we have, conserve what we have. And even though this is still beautiful, you still have this diversity. I would argue that, we're kind of missing out on these species that unfortunately disappeared. And the fauna in New Zealand, the endemic species, the tuatara, obviously, but the geckos, and even their chubby skinks, the otago skinks, they're really, really, really cool. So what I did after the Congress is that my advisor and I rented a car, Rich Galore, and we went basically around the Southern Island. And we hopped around a little bit. We kind of basically went from the East Coast to the West Coast, and then up the West Coast to the glaciers, and then back around to the East Coast, and then down. So we went, on our way, we went back through Christchurch, and then we went to the glaciers. And these are just a few images of, what New Zealand is. And this is just this amazing highway, relatively recent, where you're kind of going through this valley that splits. So apart from the northern and the southern island, you have this mountain chain in the southern island, and in a given part there's just like this very thin strait that you can go through, and you just go and you drive through these mountain chains that are still covered in green. Even though the latitude is so low, it's really amazing how humid it still is, and all the different kinds of and species that you have around us. If this, let's see if it's not, maybe it's not gonna go with the pixels because I just copied it here. See the next one. I don't know if it's my computer that's slow, or if it is that it's not loading properly, but I'll just open a new one. This is Christchurch. Let's see again. There you go. So again, you just have these valleys and then the mountain chains right around it, and you have this evidence that this is very, very recent geological activity. So I would argue that this is maybe scenically the most beautiful country I have ever seen in my entire life. You have the waterfalls going around. You do have your little kiwi holes popping up every once in a while. Fortunately, I didn't see any kiwi. I didn't go out at night to try to see one. By the way, no touching, no animals there. You're not supposed to. It's different. You can't go flipping rocks and grabbing lizards. That's illegal in New Zealand for the most part. But I would say that culturally, the country really stood out for me. It is a country that, relative to other countries, seems to be doing a good job when integrating all the different kinds of people. It does really look like a more kind of progressive country. People are very open. They tend to be environmentally friendly. This is an image from Christchurch, which had the terrible earthquake a few years back, and how they're kind of like reinventing the landscape with all these little art interventions. So kind of changing and readapting the country. So using this catastrophe, which was this earthquake, to build something new, to build something different, to build something that, you know, will take them to the next step. So this is certainly an idea that I got from New Zealand, and I feel like we do have this, like, even more now with their prime minister, with Jacinta. It's and how they are responding to the corona. It does seem to be a country that, you know, people look at it and they're like, wow, that would be an interesting place to go and to maybe live in, just because of this mindset. They seem to be always wanting to put the effort to, you know, take another step in what I believe, because this is a very particular view, I guess, but what I believe to be the right direction. Yeah, no, my next steps are, so right now I'm trying to wrap up. So as I mentioned in the presentation, I have this. So my PhD slowly developed into a little monster, but I guess that's true for everyone's PhD. And so I am trying. So we're finishing the genome of the species that I work with. I am a co-author in that, so we're trying to wrap that up. Then I have this RNA data that I'm trying to wrap up. Then I have what I think is pretty cool, which is a genomic study where we're trying to associate genotype with the phenotypes that we're interested in, which is not only coloration, that's what I talked about, but we're also interested in reproductive isolation, because you have these different populations that come in contact across the island, and they you know, produce hybrids with different levels of inviability. So that would be something else I'm looking at. Identifying sex chromosomes, because I'm interested in speciation, adaptation, traits that are under sexual selection. So right now I'm basically trying to sit down, crank some data, and write the papers, get them out. And the World Congress was this very nice opportunity to talk to some people. So I already talked to some researchers and basically said, I'm interested in your work. Will you be looking for, you know, a postdoc position? So that would be my main next step, which would be to try to find a nice postdoc where either I could be able to, you know, keep developing the system that I'm working at, which I do think that I'm not going to say only Anolis, because, you know, in Southeastern Asia, you do have Dracos, or, I mean, you name it. Color and pattern in lizards is so unbelievably diverse that I feel like this is a field that has so much potential to study, you know, this kind of approach, almost like a naturalist approach, where we can go and look at the genetics and look at the RNA, but also look at the natural history, you know, how some people are doing this great job with heliconias butterflies. There's a lot of cool stuff going on with dendrobatids now. And I would argue that, you know, squamates also do have this potential. So either do that or go work in someone's lab as a postdoc in kind of an environment that I would be able to also not only collaborate but also learn from what they've been doing. So that's kind of my 1 year plan: publish and look for a postdoc.